In this lecture, we will discuss about the basic idea of statistics that is going on in the GSEA, that is gene set enrichment analysis. In our earlier video of GSEA, we have discussed about what is GSEA and how it works, what is ranked gene list. In this example, we will understand about the statistics that goes behind this GSEA analysis. So first we will understand by the jelly beans example. In the past, there is a myth that jelly beans might cause acne. So people have tested like 20 jelly beans, for example, and they did not find any symptoms of acne. Then someone think that it could be because of different color of jelly beans. Then people have tested different color of jelly beans. Then they have found that blue color jelly beans, blue or green color. Let's stick to the story as it is. The green color jelly beans cause acne out of the 20 color they have tested. However, it only gives the probability of 1 by 20. It is 5%. 1 by 20. It only gives the probability of 1 by 20. So that means probability of 1 by 20 into 100, 5%. Probability of 5% or 0.05. So this is usually called as p-value. However, the problem with this experiment is that it has only conducted experiment on 20 samples, and they have found that there is only 5% probability that people might get acne. So in order to account for this 5%, this 5% is too high for a statistics, as some person might get acne when they have the green jelly bean, and some person might not get. In order to compensate for this 5%, what people have done, whether this green jelly bean has a significant impact in this acne, what they have done is they have done a multiple testing. That is, they have done several people's are been treated with green jelly beans. Several people's are been provided with the green jelly beans, and then now they have different sets of data, 20 like this. They have like 100 sets of 20, and now when they do the analysis, like out of this, what is the probability of the person, what is the probability that a person get acne when they have this green jelly beans. So in this case, now the data that comes out from this experiment will be much more statistically significant than the data that comes out from a single set of 20 experiments. From this, we can understand the importance of multiple testing to take care of the multiple hypothesis testing. So this is the general idea. This we will use in our GSEA to understand the basic idea of GSEA. In GSEA, there is a suppose we have to calculate the p-value or the significance of the enrichment score for a particular gene set. So gene set, suppose this gene set has like 7 genes inside it. 7 genes inside this, and it has some expression values as this data is extracted from some sample. So in order to calculate the p-value of this gene set, what happens is that whatever the expression data that this value that this gene set has, they will randomly shuffle this expression data and then again rearrange the gene based on the randomly assigned expression data, and then calculate ensemble score again. And this kind of thing can be done like for 1,000 times. If this is done for 1,000 times, then they calculate ensemble score for each time and then compare it the ensemble score with the originally assigned data. And if the original assigned then they will compare whether the percentage of the ensemble scores that is generated from the randomly assigned gene set to the actually calculated ensemble score, and the fraction of ensemble score which are above the fraction of ensemble scores of randomly assigned data which are above the ensemble score of the true data is considered as the p-value. So that is considered as the p-value of this particular gene set. However, now we have so far we have understand the idea about p-value of a particular gene set. But what about the complete gene list that we have? We also have to do multiple hypothesis testing for the complete gene list. So in this, what will happen is the same thing that is happened in the gene set, in the gene for taking care of multiple hypothesis testing for gene list. What it will do is it will randomly assign data for the gene list, and then for each randomly assigned data, it will compute ensemble score it will compute ensemble score for each gene set, and then compare it with the true enrichment score, not ensemble score. I mistakenly used the ensemble score, but it is enrichment score, true enrichment score of each gene set. And this suppose permutation is done like 1,000 times. So in that case we have for each gene set a true enrichment score and 1,000 randomly assigned gene set ensemble score. And for in order to calculate the false discovery rate, the enrichment score of a false discovery rate can be calculated by the enrichment enrichment score, percentage of enrichment score that are above the enrichment score of the gene set for a true data. That from this we can calculate the false discovery rate. So multiple hypotheses. Or gene list, complete gene list. That is also called as FDR. That is hello.
Sorry.
You need-
My mouse. Sorry to interrupt.
So for multiple hypotheses, multiple hypothesis testing for gene list L. DR can be calculated like for percent percentage of ensemble score above the true enrichment score of gene set S. This from this we can calculate the false discovery rate, and for the p-value of the particular gene set, it can be calculated like percent of enrichment score of randomly assigned enrichment randomly assigned data. Here, it is this term is important here for p-value of gene set, percentage of enrichment score of randomly assigned gene set. Assigned gene set. Above the two enrichment score of gene set. So one thing we need to understand that in this gene set we are randomly assigning only the genes that are present in this gene set, whereas in a gene list we are randomly assigning the data in the whole gene list. Okay, I hope I was able to give you some idea, if not the complete picture, what is going on in the gene set enrichment analysis. Okay, thank you.